Hey everyone, it's great to see you all. So so I'm here to introduce Yao Yuan. So Dr. Yao is a new recruit to the Department of Mechanical Engineering. She's actually a joint hire between the Department of Mechanical Engineering and the Department of Medicine Division of Cardiology. And she's actually in the Department of Mechanical Engineering. She's actually specializing in the teaching of the manufacturing program. And Dr. Yao received her bachelor's degree in Dalian Maritime University, master's degree from Tianjin University, and so that's in chemical engineering. And then she did her PhD in chemical engineering at Waterloo with Evelyn Yim, and her postdoc with Molly Choquette at University of Toronto. So so she's brand new. She just joined in September of 2024. So this is her chance to introduce her to the CBR community. So welcome, Dr. Yao.
Okay, thank you very much, Hong, for the great introduction. And it's my great pleasure to be here and meet everybody. And thank you all for coming by in such a rainy day. I'm a very new hire and I just joined in September. My research mostly focusing on biomaterial engineering and tissue engineering as well. So my lab is located, so it's part of the Center for Heart Lung Innovation and it's located in St. Paul Hospital at downtown Vancouver. So today is actually my very first official talk after I joined UBC, so it's very exciting. And I'm glad I have the opportunities to speak in the CBR seminar series. The talk I'm going to cover today is more on engineering extracellular matrix environment and how we are using those cues from the extracellular matrix environment to modulate vascular cell responses. So just a brief overview of what my lab does. So basically, we do design engineering of different types of hydrogel biomaterials. And essentially, we look at different parameters of the hydrogels, including the interface between cells and the materials. And specifically, we look at the different functional groups on the material surface and topography cues on the material surface, and try to leverage those cues to modulate cell responses to simulate cell behaviors and also try to model cell disease conditions. And also in a three-dimensional level, we also engineer hydrogels to make it a cell culture scaffold to look at the soluble biochemical molecules. And more specifically, I will talk about growth factors, how we change the concentration of growth factors and how we change the delivery of growth factors using hydrogels. And right now, what my lab is working on is to look at how the matrix mechanics are interfering with the cells and are changing the cell behaviors. And also, we're trying to develop some 3D models to simulate the disease condition in the cardiovascular field. Some of the applications that we have been working on up to now, the first one, which I will spend quite some time talking about, is the design of vascular graft. And also that one is more focusing on the blood contacting surface of the vascular graft. And we look at how the blood are responding to the surface, how the endothelial cells are responding to the surface as well. And we also use the hydrogel as delivery vehicle for cell transplantation and for protein delivery as well. And lastly, we use that as 3D scaffold to see how the cells are responding. And I'd like to start with my very first story, which is on the vascular graft design. So just a brief introduction. I'm sure many of you are familiar with vascular graft, but essentially, vascular graft is a segment of the artery that is used to bypass the blockage of a occluded artery. So in this specific diagram, I'm showing you a typical coronary arterial disease, where you will see the coronary artery are getting narrowed or getting blocked. In those cases, the downstream tissues, which are usually the myocardium, they don't receive enough blood, they don't receive enough oxygen. So in order to restore the blood flow, we are building some bypass graft to bypass the blockage and to let the blood to flow to the myocardium again. For the bypass graft, there are many different options for us. The very typical one are the vein and artery from the patients. So those are the gold standard and it works relatively well in the clinics. But the problem is that many patients, they don't have any available donor vessels. They don't have the available arterial veins that can be used as the bypass segment. So then they ended up have to go to the synthetic options, which are right now usually the Teflon or Dacron vascular graft. So those materials, they are built based on purely polymeric materials, and they work well for a short period of time. But then as time passes by, we can see those vascular grafts will start to build a lot of thrombosis and also intimal hyperplasia in the luminal surface of the vascular graft. And intimal hyperplasia is referring to the overgrowth of smooth muscle cells and deposition of a lot of extracellular matrix proteins. And you will see the big chunk of tissue that is in the luminal surface, and that will become a re-occlusion of the vascular graft. And just with those two problems, right now, vascular grafts that are purely synthetic, when the diameter is coming to lower than 6 millimeter, we have a very low success rate. So by 5 years, almost more than 50% of the graft will fail. So then my past work and has been mostly focusing on how to design a new generation of vascular graft. And we specifically focus on hydrogel materials because first of all, their similarity to the human tissue. And secondly, they have very tunable mechanical property. So the graft, the material we're focusing on is called polyvinyl alcohol, PVA. So it has this structure here. So it's a very simple structure. It has the carbon chain, and then you have the hydroxyl group. So the PVA is a very biocompatible material. It's FDA approved. And then we decide to use the PVA polymer and cross-link it, use the so-called STMP, which is sodium trimethyl-phosphate. It's a food-grade cross-linker, and it's a phosphate ring. So when it reacts with the hydroxyl group, it will go through the open ring reaction, and you can combine two polymer chains together. That's how the PVA hydrogel is cross-linked. So as you can see from the picture, this is our cross-linked PVA hydrogel. It's very elastic. It has the mechanical strength to withstand blood flow. And then at the same time, the manufacturing process is very mild. It's just room temperature. You don't need very harsh organic solvent either. And also by changing the cross-linking density, by changing the molecular weight of the PVA, we can easily tune the mechanical property to match the artery or the vein. And also one more important parameter for this PVA vascular graft is that we found it has a very low thrombogenicity. So here is how we are making the PVA into a tubular shape of vascular graft, and we implanted into rabbit femoral artery. During this 12th day of implantation, we didn't give any anticoagulant to the rabbit. And by day 12, the graft is still patent. So if there's material that are not that blood compatible, once you implant in the in vivo environment without any anticoagulant, they will get occluded very quickly. So based on our pilot findings, we think this PVA hydrogel is actually a quite promising candidate for use as a small diameter vascular graft. But then here comes the problem. So usually in the native human vessels, there is a layer of endothelium, which functions as a barrier between the blood and then the underlying extracellular matrix. And that layer of endothelium also controls the homeostasis. But then for synthetic materials, they do not have those layer of endothelial cells, but we want to achieve that endothelial cell lining on the luminal surface of vascular graft. For PVA, very much similar to any other type of synthetic material, they do not have binding site for cells. So we need to go through some modification. So first of all, we thought about maybe we use ECM protein, which is very typical. So you have a lot of binding site that the cells can bind to. But the problem of those proteins is, first of all, they have very limited in vivo stability. And then secondly, in a lot of the cases, those ECM proteins, they may non-specifically attract platelet adhesion as well. So you have a material surface that may be favorable for endothelial cells, but also prone to thrombosis. So we passed an ECM protein. And then there's the anticoagulant and heparin. So heparin has been coated to many different types of synthetic vascular graft. It reduces blood clot formation, which is good, but the problem is that it's not effective enough to promote cell growth. So then we start to look at some other molecules. And this one, which is called fucoidin, is of a particular interest to us. So fucoidan is a sulfated polysaccharide. So it's isolated from brown seaweed. It's a natural material. And if we look at the structure of fucoidan, it has very similar structure compared to heparin. We also have the sulfate group, which is at either C2 or C4. And fucoidan has been used in the cancer treatment and also wound healing. But the first reason why fucoidan attracts attention from the cardiovascular field is that fucoidan is anticoagulant and antithrombotic. And also there are some other functions for fucoidan, which is anti-inflammatory, and we also found fucoidan could modulate vascular cells. Then we start to think maybe this is a good molecule for us to modify the synthetic, or specifically the PVA vascular graft we made. We tried different approaches, and all of the approaches actually work, including coating, mixing into the base polymer, and also including chemical conjugation. And then we look at the cell responses. So here are the endothelial cell adhesion to either PVA or fucoidan modified PVA. The top row are the PVA only, and we can see at a single cell level, when the endothelial cells are interacting with PVA, they are very small, and they don't have very mature adhesion formation. So it's indicating the PVA is not very suitable for cell to adhere. And then after fucoidan modification, which is the second row, we can see the cell area become much larger. They're spreading much better, and also have those focal adhesion formation. And if we see the cells at a higher density and culture them for a longer period of time, we can see with the fucoidan modification, we can see a nice model layer of endothelial cells formed on the material surface. Versus for PVA, we only have very minimal number of cells being attached. So with this experiment, we confirmed that fucoidan is actually very promising in terms of improving endothelial cell adhesion to our PVA vascular graft. And then we move forward to test whether fucoidan will sacrifice the low thrombogenicity of PVA. And we did two major experiments. One is an in vivo blood test, and then the other one is an ex vivo blood test. So in the in vitro blood test, we do look at so the thrombin generation during a 1 hour in vitro culture. And if you look at the different groups, so we have the collagen, which is our positive control, and EPTFE, which is our benchmark material, and PVA and fucoidan modified PVA. And it's very obvious that fucoidan modified PVA didn't increase the thrombin drastically. It's remaining the similar level of PVA, and is similar to our benchmark EPTFE as well. For our ex vivo shunt result, so this is with a baboon model. And it's very similar to what we saw with the in vitro test. So after fucoidan modification, the platelet aggregation, it's towards a lower trend, and it's the same for fibrin accumulation as well. And then the last group, PVA-FG, is referring to after gamma irradiation, which is a sterilization method. But overall, we sort of confirmed that fucoidan is a suitable material to improve endothelial cell adhesion on PVA without sacrificing the hemocompatibility. But then what's more important is that we found only improving endothelial cell adhesion is just not sufficient for vascular graft. So in the case of really long vascular graft, we can see these are the main mechanism of how the endothelial cells are covering the graft surface. And in the often case, we will rely on this so-called transanastomotic growth, which essentially means the transanastomosis migration of the native endothelial cells from the artery to the vascular graft. So we're relying on those endothelial cells to migrate from here to the vascular graft. So they will eventually, theoretically or ideally, cover the surface of the vascular graft. But then human vascular endothelial cells, they have limited migration capability. So in often case, when we have synthetic vascular graft, we can only have very limited distance that the endothelial cell will cover. And then the vast majority of your graft are still having no endothelial cell coverage. So that gives us the question about how we can promote the migration of endothelial cells so that we can have a vascular graft, even when it's long, we still have the endothelial cells that is fully covering the luminal surface. And that comes to my second story, which is topography cues. So topography is something the field has talked a lot already. So if we look at the images here, the left one is the cells that are cultured on the surface with nanogratings. So the nano lines is in the y direction. And then the other image is talking about the cells that are on a planar surface, so there's no topography. And we can see with the presence of the nanogratings, the endothelial, well, not endothelial cells, but this type of cells, they can elongate and align with the direction of the gratings. And we thought about it, and maybe we can do the same thing to our PVA graphs. And we did, and we actually made some really nice surfaces with topography cues. So here's how we did it. So we have a mold that is made of elastomer, which is PDMS. And then we cast our PVA hydrogels on top of the mold and then peel it off. And in that method, we can transfer those topography cues from PDMS mold to our PVA hydrogels. And we were able to make a lot of different hydrogel surfaces that is full of different topographies. So I'm showing gratings, pillars, and lenses as well. And we can also transfer this technology to making tubular graft. So the tubular graft, we can coat the mold with a PDMS that has pattern on top of it as well. And we do coating of the mold using our PVA solution. And eventually we have a tubular PVA graft that has luminal patterns. And this is looking at how the lumen of our vascular grafts are patterned with gratings. Then we look at how the endothelial cells are responding to those topography cues. So if you look at here, I'm listing like 4 major patterns. We actually test many different topography, and these few are the ones that we think endothelial cells are responding the most. And the 2 micrometer grating, there are 2 micrometer grating, 10 micrometer grating, 2 micrometer pillar, and then convex lens. And then we can look at the endothelial cell monolayer formation by looking at V-cadherin expression. So the V-cadherin expression, so because of the presence of fucoidan on the PVA hydrogel, we can have very nice monolayer formation on all of them. But then if we think about the cell morphology, they look a little bit different among different topography. So on the gratings, we can see if we look at the grating direction and the cell direction, they're pretty much aligned. So when you have grating in this direction, the cells are tending to align in that direction, and they're elongated. Versus on the other topographies, so they're in random direction. And also the topography actually interfere a lot in terms of the morphology, the phenotype and morphology of the endothelial cells. When we have a monolayer of endothelial cells, we culture a monocyte on top of the endothelial cells as an indicator on the inflammatory phenotype of endothelial cells. And we can see the monocyte, the number of monocytes adhered to the endothelial cells are very different. If we have a 10 micrometer grating compared to 2 micrometer grating, the monocyte number actually drastically increase. If we compare pillar with gratings, the number of monocytes also increase. And this applies to the same of ICAM expression. So this is indicating to us that the topography actually can change the morphology of endothelial cells. And at the same time, they can change the phenotype of endothelial cell as well. So we decided to pick one, which is the 2 micrometer grating, because the cells are aligned very well. And also we have a low number of monocytes and a low expression of ICAM1. Something more important is that, remember, the goal of our research here is to promote migration. So we confirmed the endothelial cell can form nicely on the 2 micrometer gratings. Next one, we need to confirm they can promote migration. And we did this wound, scratch wound healing assay. This is very similar to what we would observe in terms of like vascular graft endothelial healing. So we have a monolayer of endothelial cells cultured, and we make a scratch and look at the coverage of the wound. So the top row is the M-pattern surfaces, and then the bottom row is the 2 micrometer gratings. And if we calculate and quantify the wound healing closure, we can see the 2 micrometer grating actually has a much faster wound closure. So it's indicating that the 2 micrometer gratings are promoting the directional endothelial cell migration. After confirming that, we decided to incorporate 2 micrometer gratings with fucoidan and make our PVA vascular graft. And we did the implantation in the rabbit carotid artery model. So we did this in the end-to side model, which means the end of our graft is attaching to the side of the artery. So it's more similar to the bypass grafting. And we did this surgery for like the entire month. So before the first day of implantation, we did an ultrasound check to get a baseline blood flow. And then in the midpoint, we also look at the ultrasound flow, and then endpoint, we check the flow and also harvest the graft. So this is the ultrasound result of our 2 micrometer fucoid modified graft. And we can see throughout the entire period, the graft remained patent. We have very strong blood flow going through our graft. And more importantly, when we look at endothelial cell adhesion, so the very top row is just a PVA graft with no modification at all. And we can see there's like minimal number of endothelial cells that are covering. But for fucoid modified one, we start to see more cells, but mostly are at the 2 ends. So proximal and distal are talking about the 2 ends of our vascular graft, and center is talking about the middle of our vascular graft. And then with the 2 micrometer grating incorporating with fucoidin, we can see the 2 ends has endothelial cell adhering, and then the middle also has a coverage of endothelial cells. So that's indicating by incorporating gratings with fucoidin, we can improve both the adhesion of the endothelial cells and also the migration of endothelial cells to promote the coverage of endothelium in the vascular graft. So we also did a little bit on smooth muscle cells. So this is the work we just submitted. So because usually for vascular graft, we talk about intimal hyperplasia. That's the main one of the main causes that lead to graft failure. And it's usually because the over proliferate of smooth muscle cells. And when we check the intimal hyperplasia, there's a lot of smooth muscle cells and they're proliferating smooth muscle cells. So usually smooth muscle cell has these 2 phenotypes, one is synthetic, one is contractile. So if it's a quiescent smooth muscle cells, which is in the normal healthy artery, they are in contractile. But upon disease condition or upon some activation, they become synthetic. Once they're a synthetic phenotype, they start to proliferate and then migrate. And then we thought about maybe we can use the topography to change the phenotype of smooth muscle cell as well, because we know the cells can respond to the topographies. So we did a lot of screening. I'm only showing a little bit of data here. So this is what we did. So the first thing, we are assuming that topography could make the role of changing phenotype of smooth muscle cells, but we are uncertain what topography is suitable. So we did a very high throughput screening of topography. So this is what we call a mark chip technology. So we have a little chip here. This is 2 centimeter by 2 centimeter, which can easily fit into a 6-well plate. In each individual of the chip, we have those individual fields. In each of the individual fields, we can fit into one pattern. So one type of topography. So in this example, we're fitting in 41 different topographies, and we have the unpatterned ones as a control. And we have a little bit of a replicate as well. What the topography we can incorporate, it could include gratings and lenses, holes. And here are some examples where we have hierarchical structure. You can have gratings, and on top of the grating surface, you can have a secondary topography. Here is pillars, and here is lines. So by screening all those different topography and look at how the smooth muscle cells are responding to it, we sort of found one topography, which is the 2 micrometer grating again. Interestingly, it also reduced the proliferation of smooth muscle cells. So here is the EDU assay, what we did, and the 2 micrometer grating has the lowest number of EDU. And when we look at the phenotype of the smooth muscle cells, we use the Desmin and Lamington staining. And Desmin, the more Desmin expression, it means a more contractile phenotype of smooth muscle cells. And then the 2 micrometer gratings actually increase the contractile phenotype of smooth muscle cells. And so we did a pilot animal study using the 2 micrometer gratings, which I didn't show here, but we did see some quite promising results that we don't see a lot of intimal hyperplasia formation. And then due to the time limit, I won't talk too much more about our vascular graft project, and I would like to move on to how we are engineering 3D cell culture scaffold. So similar, going back to the coronary arterial disease. So when we have the major occlusion in the artery, what's going to happen is that, so the myocardium is gonna go through necrosis. So a lot of the cardiomyocyte will go through necrosis, and then there's fibrosis, and eventually that will lead to a heart failure. So what we can do is right now there are different technologies to help with the post-myocardial infarction repair. So one is using cell transplantation. So if you don't have enough blood supply to the myocardium, we just transplant more CD34 positive cells to encourage the angiogenesis. If there's death of cardiomyocyte, we transplant more cardiomyocyte. But the problem of cell transplantation is that we have very limited number of viable cells after transplantation due to multiple reasons. One of the major reasons is that cells, they don't receive the structural support, and also there's no adhesion site for the cells to grow. And also there's the immune rejection that is killing a lot of the cells. And there's protein therapies as well. So we can introduce some growth factors, some therapeutic proteins to promote the healing of the myocardium. But the problem is it's hard to control the dose of the proteins, it's hard to control the stability of the protein, and hard to control the spatial and temporal distribution. But what we have worked on is to use hydrogel materials as a 3D scaffold to facilitate both the cell transplantation and the protein delivery. So the first work I want to talk about is our collaboration. This is the work I did when I was working with Dr. Molly Shortkit at U of T. And we collaborated with UHN, Dr. Gordon Keller's lab. So their lab has developed this PSC-derived vascular progenitor cells. So they start from the human PSC cells, differentiate, and then eventually get these vascular progenitors. But the problem is that they found those vascular progenitors, when they're being injected into the animal model, 90% of the cells are lost. And also they found if they use matrigels, it seems like it's helping with the cell engraftment and cell viability. But we know the limitation for matrigel is that it's animal source, and also there's that batch-to-batch variation. So it's not really clinically translatable. So we decided to use some other hydrogels that can be more translatable to use as matrix to deliver those progenitor cells. So we decided to use hyaluronic acid or HA hydrogels. So the reason why we use HA is that we check the cells, we look at the surface of the vascular progenitors, we do see they express CD44, which is the receptor for HA. And then we start to use the HA that are cross-linked by click reaction. So the HA are modified by either a norbornene group or a tetragen group. So these two groups, once you bring them together, they will go through the click reaction, and that's how you form a hydrogel. And then we test whether this HA hydrogel is suitable for transplantation. The first thing we did is to look at the dilation time. So we want the hydrogel to be liquid enough to go through the needle, but then we want them to fast solidify after going inside the body. And then from the graph, we can see the dilation is pretty fast. It's within a few minutes, and we can actually control the dilation time by changing the concentration of the polymer we use. And we also test whether the material can be injectable with those transplantation syringe and needles. So the needle we're using for the test is 28 G, which is a typical size for cell transplantation. And we can see as time passed, the force you need to inject those hydrogels actually increase, but it's all while below the threshold, which is 30 Newton. So we assume anything below 30 Newton, it's injectable. And then last but not least is that we found those hydrogels, when they are being stored, they don't swell very significantly. So that's very important because you don't want something that increase the volume and mass too significantly when it's being inside the heart, and that will affect the heart motion. So it looks like the physical property of the HA is very suitable for the transplantation of the VP cells. But then we want to make sure those hydrogels also have the cell binding site. And this is what we did. So we think about how we're gonna increase the not endothelial, but the vascular progenitor cell adhesion to the HA hydrogel. And we did this screening of cell adhesive peptide. So we have 24 different candidates of peptide that are derived from extracellular matrix proteins. They are short peptide, and essentially they can be modified and chemically conjugated to HA pretty easily. So we have this well plate, it's a 96 well plate. We have them coated with different peptide and then seed cells on them. Look at the cell adhesion and viability, and then screen the best peptide that can be suitable. And after we screen the peptide, we will conjugate them onto HA, and that's how we get the HA peptide polymer. And then look at the VP, we found this one peptide, which is derived from collagens, KGH-RGF. So that peptide seemed to be most favorable for the VP cells. And we incorporate it into the HA hydrogel, and we can see without the peptide, a lot of cells are dead, and then the cells are not functioning very much. But after we have 0.5% of peptide, the cells start to forming those vasculature or those tube network. And if we further increase the peptide as the material getting more and more adhesive to the vascular progenitor cells, we can get a nice model layer of VPs growing in those hydrogel. And use the same technology and workflow, we actually developed another type of hydrogel, which are modified with a peptide from laminin, and that is suitable for a PSC-derived cardiomyocyte. So this is a more sad project, but it's just proving this technology is not only applicable to vascular progenitor cells, but can be used for other type of cell as well. And then we proceed to do some in vivo experiments. So this is the first what we did. So we transplant the cells together with our hydrogels in mice, and this through a memory fed pad injection. We're using 5 million of cells, and after 30 days of transplantation, we harvest the graft. What we found is after 30 days of transplantation, we do see a lot of viable cells that are from the human PSC-derived vascular progenitors. And more importantly, we do see a lot of vascular lumens here. So we have a lot of vessels that are being formed. And if we compare our HA gel with matrix gel, it looks like the engraftment, which is this value here, the engraftment of the vascular progenitor cells are very similar. So due to the limit numbers, we didn't get a significance, but we can see the HA is seeming to have a higher chance compared to matrix gel. And also, if we count the number of vessel lumens per space, we can see they also have very similar number of vessels being formed per area. That's indicating the HA hydrogel we're using in the case of vascular progenitor cells seem to be a potential candidate to replace matrix gel. And we further refined the design of our hydrogels by incorporating some exogenous vascular endothelial growth factors. So we load those VEGF into a PLGA nanoparticle. So here is our PLGA nanoparticle, which can serve as a shell to protect our VEGF. And then after it's been implanted in the in vivo environment, it will go through degradation and slowly release the VEGF. And what we found as well in the same transplantation, there's a lot of viable cells, and also there's a lot of lumens. So what's important here is that now we're using only 1 million of transplanting cells. So by incorporating of the VEGF nanoparticles, we're able to downsize the number of transplanted cells we need for a successful engraftment. And then we move on to think about how we can deliver growth factors more efficiently. So although PLGA nanoparticle is very promising, it has been shown in the clinic to be very successful. But then the problem is that PLGA, once it's degraded, it will become acid. And it's going to create that low pH environment, which is not suitable for cells. So we start to think about leveraging the natural affinity interaction between proteins to deliver growth factors. So if we look at in vivo environment, we have the growth factor that will react reversibly with different proteins and proteoglycans as well. But if we have a hydrogel system, and we also have a partner with our growth factor and another protein that can bind to it, and by leveraging the affinity interaction, we can actually control the release of those proteins. So if it comes out from its binding partner, you will have a concentrated environment in your hydrogel. And then outside the hydrogel, you have a lower concentration. And that concentration gradient is going to drive the diffusion of your protein to come out from your hydrogel. And that's how we're doing this affinity controlled release. So the problem of this affinity control release is that deriving this protein binding partner is very difficult. So we can go through probably phage display, bacterial expression, but that will be a lot of cell culture and a purification expression procedure. So we decided to use computational tools to do this. So this is what we did. So essentially, we have some protein-protein complex that are present. You can get it from the protein data bank, or you can predict the interaction using AlphaFold. So you have two proteins, and then you look at their structure and try to isolate probably just a short segment from the binding partner. So in our case, it's the red segment here. So this red segment is the major component that is interacting with our growth factor. And by just looking at this individual segment, now it's not the whole protein anymore. It's a small molecule. It's probably just 1 kilodalton. It can be easily modified and conjugated to the hydrogel. And now we take this little sequence and then conjugate it into the hydrogel. And now it can control the release of our protein. So we don't need to go through a lot of purification process. Instead, the computer will do everything for us. And this is an example of how we did it. So we still are looking at the VEGF, vascular endothelial growth factor. And this, we're deriving the short peptide from bevacizumab, which is an antibody for VEGF. So this is what we got for the VEGF and bevacizumab. So the center part is VEGF, and then the two ends are the antibody. So we have this complex, which is very complicated and has many chains. We try to clean it up a little bit to leave only one subunit from VEGF and one chain from the binding partner, which is the bevacizumab. And then what the software do, which is online server, is available for everybody to use. So what it doing essentially is that it will set a scanning window. Scanning window means it's 5 amino acid, 10 amino acid, or 15 amino acid. And it will go through your chain from one end to the other. And each time you go through it, the sequence, the 10 amino acid sequence will be isolated. And then the computer will calculate its interaction between the short segment and your VEGF. And it's based on energy. And usually what the software gives you is this plot. So the y-axis here is talking about the energy you required to bind those two chains together. And the lower, it means the easier to bind. So it's more favorable for the binding between your segment and your protein. And after this whole scanning through your protein chain, what you will be able to have is the lowest point, which is the most significant segment that will contribute to the binding to your protein. And in our case, is this point. So it's around, so start from the 99 amino acid all the way to 108 amino acid. And if you look at the structure, it's this red part here. So with this platform, what we can do as well is to look at how we can change the peptide length. So what we did, so what I showed in the previous slide is using 10 amino acid. Now you can use 5, you can use more, you can use like 57. And you can see the binding affinity actually change based on the length. And depending on what affinity you're targeting, you can select different lengths of your amino acid. And what you can also do is to identify the hotspot residuals. So which amino acid are contributing most significantly to the binding. And then you can do some mutation to adjust the affinity as well to release your protein. And we decided to proceed with our 10 amino acid sequence, and we check the binding. We do see a quite nice binding. So this is the binding and unbinding. And we got a dissociation constant, which is the binding affinity between our peptide and the protein is around 46 micromolar, which is acceptable range for our affinity control release. And we did something similar to PDGF, which is platelet-derived growth factor, because we know that usually when we want to have a mature vascularization, we want the VEGF to be present and promote the proliferation and migration and angiogenesis of endothelial cell first. And then we want the PDGF to come into play to attract parasite or smooth muscle cell to stabilize the vasculature. So what we did here is that we try to have a system to deliver VEGF first and then deliver PDGF, so that we can have the system to promote endothelial cell angiogenesis first and then the PDGF to stabilize, deform the vasculature. So we need to have a PDGF that, a PDGF binder that has a stronger affinity to PDGF, so that the interaction is stronger and then the release is slower. And this is what we got. We also got a sequence that has a dissociation constant, which is much stronger compared to VEGF. And when we do the cumulative release, we can see now the VEGF actually released pretty fast and it pretty much plateau after 6 days. Versus the PDGF will slowly release in the beginning and eventually come out more and more as we extend the release time. So that is how we actually deliver the whole system to try to control the concentration and the presence of different growth factors to support our vascularization process. And with that, that will be the last project I want to talk about. And I would like to acknowledge many of my previous co-workers, my previous labs from U Waterloo and also U Toronto and our collaborators. This is a good reminder. I haven't got a picture of me and my own lab. Maybe I should get one as soon as possible. But with that, thank you very much for your attention, and I'm very happy to answer any questions.
Yes, Cedric Carter. Can you hear me?
Yes.
Okay, super. So we had an interest in post-stent stenosis. And the theory was it was platelet-derived growth factor. And what we tried to do, and it didn't really work out very well because clinicians weren't very cooperative, we tried to sample platelets for activation, CD62, the activation marker, pre and post-stent. We got some preliminary data. The thing rather died on the vine. Is PDF a component of your hydrogel-related stenosis?
Are you talking about the PVA hydrogel? Is that suitable for the stent coating material?
Yes.
Okay, so when we tried the plain PVA without any modification, we did look at the platelet activation. We have very limited number of platelets that are growing on top of PVA. And when we check the microparticle generation, we have also limited number of microparticle generation from the platelets. So it means, so the PVA should be something that is more favorable if you are trying to prevent platelet aggregation or platelet activation. So I think that's actually a promising material. But for stent, something I think we need to explore a little bit further is, so when we have the hydrogel coating on the surface, whether we can have a very nice hydrogel being formed on the surface, and when you expand the stent, it's not going to cause any rupture of the hydrogel, because PVA is rather a stiffer material, because when we use it for a vascular graft, we require it to be super elastic. But for stent coating, I imagine we will need to tune the mechanical property quite a lot.
Right, and in terms of, did you look at something like Tigrelor or one of the strong antiplatelet agents in your systems or not?
Sorry, I didn't hear very clearly.
Sorry, beg your pardon. Well, you mentioned heparin and graft stenosis and thrombosis. Most of the interest is really focused on antiplatelet agents. Tigrelor is like a super aspirin. Have you looked at that at all in any of your systems?
I didn't hear the very last part.
Oh, sorry. Have you looked at any of the antiplatelet agents in your systems in terms of platelet adhesion activation? Specifically Tigrelor, it's a proprietary one. It's like Plavix.
So did I do any platelet? So we did use the PRP test. I probably didn't show the SEM here. So in this experiment, we did for the in vitro blood test, we collect the platelet-rich plasma, and we culture the platelet-rich plasma with our PVA hydrogels and also fucoid-modified PVA hydrogels. We look at the number of platelets being attached to the surface and also the morphology of the platelets using SEM, and also flow cytometry to look at the CD62P expression as well.
Okay, thank you.
Yeah, no problem.
Nice talk. Thank you. Maybe I have two questions. One is both on cell transplantation work you did. So those you showed kind of cells are surviving in the mice. do you know the functional activity, functional recovery of those functional activity of the cells? Is it preserved or is it how it is?
That's
And, yeah, so maybe second part is kind of like, is it a syngeneic transplantation or is it an allogenic transplantation? So I'm looking for the what kind of like the cells are actually used. If it's a different strain of the mice or different type of cells actually used there, because in there actually there is no immune mediated kind of like toxicity or kind of clearance. Is it playing a role there? That's what I'm looking for.
I see. That's a very good question. I think I can answer the first question. The second question, we're collaborating with our another group at UHN, and I believe their mice, I don't think they did anything to the mice. So it's just regular mice and wild type mice. And then they just directly inject the cells and the hydrogels into the memory fat pad without any pretreatment. The first question, sorry, what's the first question again?
The first question is the functional activity of those cells.
So the functionality, we're only looking at whether those cells can form vasculature. So those lumens are something we're looking for because we want those CD34 positive or the vascular progenitor cells to form new vasculature. So that's the only thing. But whether those vasculature can function very normally like the capillary in the native environment, I think that will require more animal models to test. Right now, it's just a very pilot study to confirm those hydrogels can promote the engraftment of the cells.
Thank you. Maybe like one last question. Since you're starting the lab in UBC, actually, like could you a little bit tell about what are the exciting projects actually you're doing here in UBC?
So we do something that is more as follow-up research here. Right now we know that Endothelus fucoidan actually it's a molecule that can promote the healing of endothelial cells. So instead of using a vascular graft, we're trying to explore drug-eluting stent. So drug-eluting stent, because we know the drugs that are being eluted can suppress smooth muscle cell proliferation, but then at the same time, we want to make sure there's endothelial cell healing as well. So we want to look at whether the fucoidan can be used for that purposes as well. And then another approach is that we have many different hydrogels, which is something I didn't talk too much. We found for the hydrogels, we're using as cell scaffold, not just the chemical properties that make a big role. The mechanical property also change the microvasculature formation. So if it's too stiff, then the microvasculature won't form very well. It's rather softer, then they will start to migrate and sprout. So we're looking at developing different models to look at how the matrix mechanics are interfering with the endothelial cells and contributing to the microvessel formation.
I'm not concerned, but I'm curious really about some of the stabilities of these hydrogels when you leave them for prolonged periods in vivo, enzymatic breakdown and so forth, especially things like hyaluronic acid and that.
I think for hyaluronic acid, it will definitely break. I think what we have looked at is up to 1 month, and after 1 month, a lot of probably 90% of the hydrogel will degrade. And for our purposes, the reason why we use HA, because it's a natural material and it can degrade. We don't want a synthetic material to stay in the body permanently. So as the reason why we're including cells and we're including proteins there is to, as the material degrade, we're encouraging the native tissue to replace it. So we're promoting the cells and the vasculatures to grow inside the hydrogel. And then eventually you will have full integration instead of having an external material to be permanently stable in the body. For the PVA, on the other hand, we don't see it to degrade very significantly. We culture them in buffer with BSA for like up to three months. We don't see any weight loss. And in our in vivo study, that's up to 1 month, which is a relatively short period of time. But then when we take out the vascular graft, we check the luminal surface, there's no damage. If we have pattern, the pattern are still there. So there's no any significant sign of degradation.
Thank you very much.
Hi, Dr. Yao, it was a great talk. Thank you. So I wanted to ask a follow-up question on your grading patterns. You mentioned in your talk that you observe like a lot of monocyte recruitment when you have like 10 micron size grading patterns, and it's less in the 2 micron. I was wondering if you have observed any cell stress, cell death, and in your in vivo experiment, do you have any cell death because of this grading pattern on the vasculature?
So for the grading patterns, because we do see that the 2 micrometer grading seem to have less number of monocytes adhere to the endothelial cells. So we only select 2 micrometer gratings for our in vivo study. In terms of how the 10 micrometer grating will perform in vivo, we don't have a clear idea. We haven't done any in vivo study. And for your question about the cell death, we actually, because we are relying on the native endothelial cell to cover our vascular graft, we are not like having vascular graft that are pre-stated with endothelial cells and then implanted in the rabbit. So it's hard for us to judge whether there is cell death because of grating or not. Essentially, what we saw on the luminal surface are alive cells because they have to be alive to come and migrate to the vascular graft.
Yeah, thanks for your talk. I'm curious why you did your vascular grafts on rabbits in the first study. Is it just that the vasculature is bigger and it's easier to do the transplantation, or is it similar to humans compared to mice?
I think, of course, the size is a big issue. When we think about the fabrication, the smallest vascular graft we can fabricate with PVA is 1 millimeter. And for mice or rat, it's probably too small as a coronary artery, bypass artery, and rabbit is most suitable. And before we move on to large animal studies, we want to go through a small animal to make sure that this is possible and this is promising. And for the rabbit, the distance we can make is also longer because the rabbit neck is longer, and we want to make like 2 centimeter graft. Compared to the other animals, we probably can only limit to 5 millimeter-ish. And with a longer graft, you can better see how the endothelial cells are migrating through the lumen of the vascular graft.
Great, thank you. Yeah. Right. Any other questions in the room or on Zoom? Okay, well, that was fantastic. Thank you so much.
Thank you, everyone.